So thank you all for coming. So I'm fascinated by this question of what triggers adaptive radiation, like the spectacular adaptive radiation of Malawi cichlids. And we think we know the answer to this question, right? It takes ecological opportunity, so lots of resources in a new environment with few competitors. This creates an environment where frequency-dependent disruptive selection among these individuals drives multiple fitness peaks on the adaptive landscape. And third, mechanisms like hybrid swarm can provide a source of adaptive alleles and also trigger adaptive radiation. And finally, aspects of genetic architecture, like large effect alleles, can facilitate diversification. So for this talk, I'll first introduce the San Salvador Island adaptive radiation of pupfishes and argue that they're a spectacular example of novel ecology, rapid trait diversification. And at first inspection, it looks like there's apparent evidence for all four of these theories or mechanisms of adaptive radiation. But on closer inspection, I'll argue that there's minimal evidence for all four theories. And to give away the punchline, at this point I have no idea what's triggering adaptive radiation in this system. But I think that the answer is actually much more complex and fascinating than any of these single factor explanations that I've just introduced. So, the pupfishes. San Salvador Island is a tiny island in the middle of the Caribbean with these salt lakes in the center of the island here. And so to orient you, here's what the salt lakes look like. Macroalgae dominated habitats where all three pupfish species coexist in this habitat. So we're not talking about habitat divergence here. This is trophic divergence. So there's a generalist species on the top, this snail-eating species, and finally a scale-eating species. And just to give you a sense of the complexity of these phenotypes, I'm going to pass around now these 3D printed skulls, as well as the actual specimens, which are unfortunately only this big. So, the scale eater, here it is in the wild, in the red circle, bites scales off of neighboring pupfishes all day long. So, frozen here. Here we go. So, I always show this video because it's so entertaining, but here it is in the lab. So here's the scale eater, biting scales off a euthanized generalist. Look at the spray of scales off the side of that fish. So this makes up 50% of their diet. And, let's see, I'm going to show it again. And just to put this novelty in context, there are other scale eating specialists out there, but the most closely related extant scale eater is actually an African cichlid, separated by 168 million years of evolution in between. And throughout here, there's actually over 6,000 fish species that have failed to specialize on scale eating. So that to me is sort of the epitome of ecological novelty. Now the second specialist on San Salvador is a snail eater. It actually has this novel nasal protrusion, here's the upper jaw, that's actually a skeletal protrusion, as you'll see on those 3D prints. And in the lab, here it is feeding on a crushed snail shell, but I think this nasal protrusion stabilizes the shell and enables it to exhibit greater crushing ability. But again, there's another specialized molluscivore out there with a nasal protrusion, but distant related, Aspera off the coast of South Africa. So, novel trophic specialists, I hope I've convinced you. But what about that hallmark of adaptive radiation, rapid trait diversification? So here, across the Caribbean phylogeny of pupfishes, I'm showing the distribution of jaw diversification rates across that as a heat map. And here, just note in blue are neighboring islands. The lineages leading to those scale eater populations on San Salvador are actually diversifying about 1,000 times faster than neighboring populations. And we can also look at these traits across the heat map, which I'll call a morphogram. So for each trait that I've measured, the heat color indicates the diversification rate on San Salvador relative to background rates. And again, just note that this diversification is concentrated in the oral jaws, corresponding to the massive oral jaws of that scale eater. And finally, if we model this as an OU process instead, using the Bayou package, we can see that these three scale-eating lineages are being pulled towards a new adaptive zone, a new fitness peak, indicated here by the red. So, a classic adaptive radiation, even though it's only 3 species, I would argue. So what about ecological opportunity? And indeed, there's actually only 2 places in the entire range of pupfishes across the Caribbean where we find these sympatric adaptive radiations of trophic specialists. And both of these places are large, shallow, salty lakes with few competitors. In fact, in both cases, there's only a single competing mosquito fish species. So that's it. I should stop right here. We've got our explanation, right? Just like Lake Malawi, we have a large lake, few competitors. This is driving adaptive radiation. And in systems like Lake Malawi, where there's no other replication or where the adaptive radiation occurs everywhere, we can't really ask about environments where we have a generalist lineage where adaptive radiation has not occurred. But in pupfishes, we can. So if we go to neighboring islands across the Caribbean and the Bahamas, these also have saline lakes with generalist pupfishes, but no specialists. And here on the right are neighboring salt lakes with just generalists. Here are 3 different salt lakes on San Salvador that support generalists and specialists. And I presented in evolution in the past that when we compare these environments, they're remarkably similar. So one other mosquito fish competitor, similar lake areas, similar genetic diversity across these generalist populations, morphological, dietary, isotopic, and stomach content diversity across all these generalist populations, suggesting very similar ecological opportunity. In fact, the only difference I've found, in a few lakes on San Salvador, there's more macroalgae species, but they're extremely rare. I don't think this is the trigger. So, minimal evidence for ecological opportunity driving adaptive radiation in pupfishes. What about the second mechanism of negative frequency-dependent disruptive selection due to competition and multiple fitness peaks? Now again, as I've shown you in the past, I was able to raise over 2,000 F2 hybrids of the specialists, generalists, including back crosses, bring these back to San Salvador Island in my checked luggage, 2 different low and high density field enclosures in 2 different lakes. And then measuring 16 different traits on these hybrids, we can construct a fitness landscape for that environment in San Salvador. So here I'm plotting survival probability of these F2 hybrids relative to their position in a phenotypic morphospace measured from those 16 different traits. So essentially this just shows the similarity of each hybrid to the parental species. The generalist here, which sits on an isolated fitness peak, separated by a shallow fitness valley towards a higher fitness peak corresponding to the snail eater. And finally, this scale eater is isolated by a large fitness valley. And I haven't found the fitness peak for the scale eater yet. I think it might be out here. And by, by varying competition, by changing density between these field enclosures, I've shown that in the high density enclosures, the fitness landscape is multi-peaked. So this suggests competition is driving multiple fitness peaks. But everything I've shown you so far is just a 2-dimensional, multivariate summary of those 16 traits. And let's see, this keeps going backwards. Here we go. So, but what about the interactions between pairs of traits and fitness? So here, just to give you a sense of the complexity of that full fitness volume, this is 6 different functional traits: oral jaw size, eye width, nasal protrusion length, and body depth. And pairwise interactions with fitness. And I'm only showing you the nonlinear interactions here. But it goes from everything from flat to 1 peak to 2 peaks to even 3 peaks. So for example, lower jaw length, when it interacts with upper jaw length or nasal protrusion, is a single fitness peak landscape. But its interaction with body depth may be as much as 3 fitness peaks. So in conclusion, competition does drive multiple fitness peaks, but only in some trait dimensions, subsets of that 16-dimensional space. So what about the second aspect here of negative frequency-dependent disruptive selection? So we can get at this question by looking at the residual survival probability of the survivors residual from this fitness landscape, which is just based on their phenotype. So here I'm showing you the residual survival probability of the hybrids relative to the phenotypic distance to the 10 nearest neighbors in that morphospace. So this is an index of the frequency of competitors within the local region of that morphospace. Now here, look, as we go this short distance here, we're reducing the number of competitors, so more phenotypically similar competitors. We see exactly what we predict, which is that negative frequency-dependent disruptive selection, right? So the fitness of hybrids increases as we decrease the frequency of competitors. But then we get past this region here in both lake environments, and the fitness of hybrids plummets as we get to extreme hybrid morphologies, which I think means that absolute performance dominates at these larger scales. So essentially, if your hybrid phenotype is so extreme that you can't survive on any resource no matter what you do, it really doesn't matter what the frequency of your competitors is. And looking at the scale of this, so this is a distance to about within one sort of standard deviation, on the fitness landscape, the scale of that negative frequency dependent relationship is within a fitness peak. So I speculate that negative frequency dependent disruptive selection acts within the scale of populations, but that performance is actually determining the broader landscape, this fitness landscape topology. So performance may be shaping these multiple fitness peaks. So no evidence. What about hybrid swarm? Now initially we can construct a fitness tree for Caribbean populations of pupfish, and the prediction of hybrid swarm is that you have introgression into San Salvador Island that spurs adaptive diversification. And we see significant evidence of that asymmetric gene flow from some of these islands into just the specialist populations on San Salvador Island. Again, I should just stop right here and say, look, hybrid swarm, it's triggering adaptive radiation. But on closer inspection, if instead we look at a population graph or phylogenetic network of Caribbean populations using tree mix, so here in black is the maximum likelihood phylogeny. But all these orange and yellow arrows are indicating gene flow among different islands in the Caribbean. And the models that fit the data best include up to 20 different migration events among islands, suggesting that there hasn't just been asymmetric gene flow into San Salvador, but everywhere. Essentially just that gene flow is pervasive throughout the Caribbean. Skipped ahead there. So hybrid swarm, yes, but it's everywhere. I don't think it's the trigger. So what about this final aspect of genetic architecture? Oh, I'll go really quickly. So we've started to do PGL mapping here. Again, it's concentrated on the oral jaws, where you see this darker red coloration has larger effect alleles. So 4 different aspects of the oral jaws explain up to 15% of the variation in jaw length. And this is actually correlated with trait diversification rates, but only for the scalier traits. Nasal protrusion looks like all small effect alleles. So in summary, subtle differences in ecology, local scale of frequency dependence, hybrid swarm is everywhere, and large effect alleles for scale leading, but not snail leading. But I also want to give you a preview of our upcoming work. My grad student, Joe McGurr, has been sequencing 35 whole genome sequences, 6x coverage, and he's found another surprising result. So by focusing just on fixed regions strongly associated with jaw length, he found 4 different regions with candidate genes, very good resolution of 50 kb here, and just a handful of SNPs within these regions that are fixed between specialists. But we see no depression in diversity within these regions, no evidence from Chichima's D that there's been hard selective sweeps, which for a novel ecology like this of scale leading, I was expecting hard selective sweeps. Instead, it looks like standing genetic variation. So in summary, we have similar ecology, standing genetic variation for large effect alleles. What's going on? And partly it's stabilizing selection. But I think it's also this aspect of why haven't they escaped? And this is hand waving, right? Why don't the standard theories apply to pupfishes? And I think part of the answer is that when we're talking about novel ecology, different rules apply than when we're talking about local adaptation or habitat divergence. My best guess at this point is that epistasis among large effect alleles that exist as standing variation is further constraining the evolution of scale leading, maybe similar to Lenski's long-term evolution experiment. Thank you.